Hello students, today's lecture is on Brucella ring test and mastitis, included in the chapter of microbial analysis of milk. Brucellosis is a primarily disease of dairy cattle which is caused by a organism Brucella abortus. It is a non-spore bearing, non-motile, aerobic gram-negative organism of alpha proteobacterium which is included in the family of brucellosis. In case of brucellosis, the infected cows excrete the causative organism, that is Brucella, in milk, from where the organism can be isolated directly by plating onto the surface of the plates of serum dextrose agar containing various antibiotics to inhibit the growth of contaminants. Or the presence of Brucella in milk can be detected by a milk ring test or Brucella ring test, which is a rapid screening method. Milk ring test or Brucella ring test. Milk of cows suffering from Brucella infection of the mammary glands may contain Brucella agglutinins, that is, antibodies. When concentrated suspension of organism, that is Brucella abortus, stained with hematoxylin, is added to the milk sample, the bacteria, that is antigen, are agglutinated by the antibodies which are present in the milk and rise up with fat globules to form a deep blue color ring in a cream layer. This test is very sensitive and may apply to bulk milk, since a positive milk continues to give a blue ring reaction even when that milk is highly diluted with negative milk from non-infected cows. Positive result may obtain if animal is immunized against brucellosis. That is false positive test. So procedure: pour 10 ml of milk in a test tube. Then add a drop of stain antigen and mix thoroughly by shaking. After mixing, incubate the tube in a water bath at 37 degrees Celsius for about 40 to 50 minutes and then observe for the formation of a blue color ring at the top of the milk surface. In negative control samples, there are no agglutinins in the milk, so no agglutination. Then there is a white cream line and the rest of the milk turns to blue due to stain antigen. Result interpretation. A strongly positive reaction is indicated by the formation of deep blue ring on top of white column of milk, which indicates the presence of high concentration of agglutinins in milk. The test is considered to be negative if the cream layer of the milk remains white while milk column turns to blue, while the 1 plus, 2 plus or doubtful result depends on the intensity of the color in cream layer of milk column. Next to milk ring test or Brucella ring test, the another example of udder infection is mastitis. Mastitis is defined as it is an inflammation of the udder which is of microbial origin and is the outcome either of a local or of a general infection. The common symptoms of mastitis includes the inflammation of the udder, swollen, hot, red and painful udders. Mastitis can be classified depending upon the symptoms. For example, acute or clinical mastitis, in which the macroscopic changes in the milk or udder are readily detectable. Subacute or subclinical mastitis, in which milk of the mammal and udder it appears as a normal and it can be diagnosed by examination of milk sample for various compositional changes and the presence of pathogenic bacteria and presence of somatic cells in milk of that mammal. Chronic mastitis, in which there will be a little compositional changes in milk and almost complete absence of pain in the udder of the mammal. Depending upon the causative organism, mastitis can be classified as a contagious mastitis, which is caused by pathogenic bacteria, that is Streptococcus agalacti. Common mastitis caused by Streptococcus, Staphylococcus or E. coli, and summer mastitis, which is caused by pathogen, that is Corynebacterium pyogenes. The milk of the animal suffering from mastitis is called as a mastitic milk. Mastitis is also known as a mammitis. Next is changes in the composition of milk which is associated with mastitis. In mastitic milk, the percentage of solids, fats, lactose, total proteins is decrease, but there will be increase in the concentration of sodium and chloride content of milk. Next, detection of mastitic milk. Presence of mastitis can be detected by using a pH indicator dye, that is bromothymol blue or bromocresol purple. Mastitis can be detected by the bromothymol blue test, which is based on the fact that milk from infected udder is usually alkaline due to the presence of blood cells in the milk, and which results in increase in the pH of the milk as high as 7.4. The pH of normal milk ranges in between 6.7 to 6.9. But when the animal is suffering from the mastitis, which is a udder infection, then the milk of that mammal will contain the blood cells, and due to which the pH of mastitis milk is increased. And this increase in pH can be detected by using a pH indicator. So add 2 to 3 drops of 0.9% of bromocresol purple solution in 3 ml of milk and observe the change in the color of the solution. The normal milk after addition of solution will appear as yellow due to its acidic pH, while mastitic milk will appear as blue or purple due to its alkaline pH. The second test which is used for the detection of mastitis is called as a California mastitis test or CMT. The reagents which are used for this test includes sodium hydroxide, then TPOL, bromothymol blue, that is the pH indicator. All these are added in distilled water. So this test is based on the presence of increased number of leukocytes and increased alkalinity of the milk sample. This is because of due to the presence of udder infection, which includes the presence of leukocytes and increased content of basic salt, that is increase in pH. So in procedure, milk is stripped directly into the chamber of plastic paddles with 4 chambers. So the paddles having 4 chambers. In that chamber, test reagent is added in the same ratio to each chamber. Then the milk and the reagent are rotated by movements of the paddle, and the reaction is observed immediately. The positive milk sample will turn to greenish blue due to its alkalinity, and due to the presence of increased number of leukocytes, a precipitate or gel is formed which indicates the positive test, which indicates the presence of mastitis. Next test which is used for the detection of mastitis is catalase test. This enzyme is present in every living cells. Leukocytes contain increased quantity of this enzyme. The determination of catalase will give fair indication of the presence of leukocyte in milk sample. In udder infection, the number of the leukocyte is increased in the milk. Therefore, the determination of catalase will indicate about the presence or absence of infection. The amount of catalase present in milk sample is determined by the ability to break down this hydrogen peroxide. So, catalase enzyme acts on hydrogen peroxide and convert this hydrogen peroxide into oxygen and water. So, 1% of hydrogen peroxide is used for catalase test. So, appearance of the oxygen bubbles after addition of 1% hydrogen peroxide solution in milk indicates the test is positive, which indicates the presence of leukocyte in the milk, that is the presence of mastitis. Chloride test. This test demonstrates the presence of increased quantity of chlorides in mastitic milk. In this test, two solutions are used. Solution A, which contains silver nitrate, and solution B, which contains potassium chromate. So, in procedure, take 1 ml of milk sample in a test tube. Then add 5 ml of solution A, which contains silver nitrate, and then add 2 drops of solution B, which contain potassium chromate. After addition, mix this reagent in milk properly by inverting the test tube, and then observe the development of yellow color. A yellow color indicates the presence of more chloride contained in the milk, while the development of brownish or red color, which indicates the less chloride contained. So, the presence of brownish red color indicates negative result. Simplified Resazurin Renate test. This test is simple and it is used for the detection of bovine mastitis. In this test, a sterile test tube is placed with a tablet of Resazurin and renate, and then add 10 ml of milk in a test tube. After addition, incubate the tube at 37 degrees Celsius for 1 hour. After incubation, the change in Resazurin color and the time which is required for the coagulation are observed. Resazurin is a redox indicator which changes its color after reduction, while renate is an enzyme which is responsible for the process of coagulation. Delayed coagulation and faster resasurin reduction indicates the presence of mastitis. The coagulation of milk by rennet is slowed down due to disturbed salt balance in milk and the presence of large number of leukocytes in milk reduces resasurin dye faster. So delayed coagulation and faster resasurin reduction indicates presence of mastitis. The presence of mastitis can also be detected by detecting the number of somatic cells present in milk sample. Somatic cells are the cells which consist of white blood cells and epithelial cells. Somatic cell count is the indirect test which can be used for the determination of quality of milk. Somatic cells are always present in milk, but the number of these somatic cells increases when there is a presence of mammary gland infection. When udder are healthy, the somatic cell count in milk ranges in between 50,000 to 1 lakh cells per ml of milk. If the somatic cell count is greater than 2 lakh cells per ml, then it indicates the presence of udder infection, that is mastitis. The presence of high number of somatic cells in milk reduces the quality of milk and milk products, and it also affects the shelf life of milk and flavor test. Somatic cells can be estimated by using sodium lauryl sulfate test. It is a presumptive determination of somatic cells and the severity of mastitis is based on increase in viscosity of milk on addition of sodium lauryl sulfate solution in milk. 2 ml of milk is mixed with 2 ml of sodium lauryl sulfate reagent. After addition, shake it for 20 seconds in a tube and then observe for coagulation of leukocyte. After shaking, if no viscous layer is formed, which will indicate that milk contains less than of 1 lakh of leukocytes per ml, which indicates the absence of mastitis. If slight viscous layer appears, then it indicates that milk contains a leukocyte in between 1 lakh to 5 lakh, that is subacute mastitis. If central viscous cone disappears after stopping rotation or after stopping shaking, then the leukocyte count will be in between 5 lakh to 50 lakh cells per ml, which indicates presence of acute mastitis. And if the leukocyte count is more than 50 lakh, when the central cone persists, then it indicates the presence of acute mastitis. So references for Brucella ring test or milk ring test and for mastitis, a book of A Comprehensive Dairy Microbiology by J.S. Yadav. Thank you.